Hello everyone. This is pathogenesis presentation. This is also made by a student. So first of all, what is pathogenesis? This presentation made by Gavit Aryan Kumar. So pathogenesis, pre-penetration, penetration, post-penetration. Pathogenesis is the process of infection or the actual way in which the disease develops in plant body. Infection is the establishment of a pathogenic microorganisms within the host. It signifies the sum of biological processes which take place in the host body after penetration of the pathogen independent of the fact whether the pathogen causes a disease or not. The potential capacity of infection of any pathogen is called its pathogenicity. The pathogenicity of every pathogen is its specific feature. The degree of pathogenicity known as virulence. Pre-penetration. The pre-penetration phase includes the growth of pathogen before actual entry or penetration into the host. Spore of various pathogenic fungi germinate on the surface of the host. During germination, metabolic activities of spores increase considerably. Spore germination besides various physical factors like moisture, temperature, light, pH, oxygen, carbon dioxide, etc. During germination, the spore produces filamentous germ tube from one or more germ pores. When more than one germ tubes develop, one of them grows more rapidly than the others. The formation of germ tube is affected by environmental factors and susceptibility of the plant. Penetration. Spores show various penetration mechanisms. The infection threads of pathogenic fungi may enter the host through natural openings, stomata, lenticels or hydatodes, through wounds or direct penetration. Most of the rust and downy mildews enter through stomata. When germ tube reaches near a stoma, its terminal part swells to form a vesicle. This is known as appressorium. Most of the protoplast of the germ tube accumulates in the appressorium, which is separated from the germ tube by a septum. The content of the appressorium pass in the vesicle. One or more penetration hyphae grow from this vesicle and they form inter or intracellular mycelium. Post-penetration. The post-penetration phase includes the development and growth of the pathogen after penetration. In this phase, colonization of the pathogen takes place. After successful penetration inside the host, the pathogen secretes several types of substance such as enzyme, toxins, or growth regulators. These substances physiological and morphological disturbance in the host plant. A cellulose wall occurs around the plant cell, and it is necessary for the pathogen to break this wall to seek entry into the host. Various types of enzymes like cutinase, pectinase, etc. secreted by the pathogen carry this work. Some pathogens also secrete certain substances which protects them from the lytic enzymes produced by the host. Besides enzymes, several pathogens secrete certain harmful substances known as toxins. Toxins differ from enzymes in that they do not attack the structural integrity of tissues. Toxins affect the metabolism in a subtle manner and act directly on the protoplast of the cell. Thank you very much. Hello everyone. This is Pathogenesis presentation.